The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 12 of eLife's podcast, produced by The Naked Scientists. I'm Chris Smith. This month, does eczema cause or prevent skin cancer? How does the bug behind sleeping sickness sidestep the immune system? And Nobel laureate and eLife editor-in-chief Randy Schekman explains why some science publishers are distorting the system. First though, some neuroscience to get your teeth into.
My name is Fan Wang, and I work at Duke University Medical Center in the Department of Neurobiology. So the question we had in mind are, how do we use our muscles in our face and mouth in a highly coordinated manner? For example, when you eat banana, your tongue is trying to position the banana between your teeth, and your teeth is chewing it down. And once it's grinded into a mushy bolus, you're automatically using other muscles in your throat to swallow it without much effort.
And I suppose that the additional complexity is that these are muscles that are paired, because you've got one set on one side of your face, one on the other, and they've got to move together in order to move your jaw symmetrically.
Yes, exactly. So mammals have their jaws, a lot of them fuse in the midline. So if you try to chew on one side, the other side automatically also move together.
So one therefore has to ask, do they work in that coordinated way because there are connections between the nerves that supply those individual muscles at the level of the brainstem where those motor nerves originate? Or are there signals coming down from above, higher centres up in the brain, which are activating them together so that you get a coordinated movement?
Precisely. So we actually took a relatively naive approach. We wanted to see what neurons are connected to the different motor neurons. So we use a virus that can infect from the muscle and to the motor neuron that sort of supplies the muscle, control the muscle. And then the virus can replicate itself in the motor neurons, then jump to other neurons that connect to these motor neurons. So when we did such studies for the jaw muscles and the tongue muscles, when we labeled the neurons connected to one group of motor neurons, we find those neurons also send axons to another additional group of premotor neurons. And what we find are the premotor neurons have one branch connected to the left side of the jaw muscle, the other branch connect to the right side of the motor neurons that control the right muscle, thereby providing a simple solution to synchronize the left and right side.
So does this mean then, if I were to go in and deactivate those premotor neurons, which are effectively the coordinators of this movement, on one side of the brainstem, just if I deleted them or took them away, animals should still be able to activate the muscles coordinately on both sides of their face, because the other side would nonetheless still turn on the right groups of motor neurons in the right order?
Exactly. I think that's our prediction, and we're actually trying to do the exact experiment that you just proposed.
Probably the most important question of all, have you discovered why we do or don't bite our tongues? It sounds like a slightly facetious question, but actually it's an important one, because tongue movements are very highly coordinated with chewing movements, aren't they? Because otherwise we would be lopping off our tongues every time we had a meal.
Exactly. There are two aspects. Neurons that control the motor neuron or tongue sticking out are always simultaneously control the jaw opening motor neurons. In a sense that when you're sticking your tongue out, you're obligated to open your jaw. And that is the simplest mechanism to ensure you don't close your jaw but bite into your tongue.
What have you actually found, though, that is genuinely new here? Because when one goes and looks at a patient who's had a stroke, medical students and doctors look at the patient and ask them to, say, raise their eyebrows or smile, because this can distinguish between whether they've got damage to the lower motor neuron or the message not getting through from above. And it's well known that if you damage the brain stem, the motor neurons, you will be paralysed on one side of the face. But if the upper motor neuron is damaged, you can still nonetheless make these symmetrical bilateral movements. Good
point. Yes, so especially in the brainstem, it seems the upper motor neurons, a lot of them do have bilateral projections, although with a ipsilateral or one side sort of a concentration, a bias. Previously, people find you can stimulate one side of the cortex, this is done in animals, and then you can initiate bilateral synchronized movement of the jaw. However, when you cut the midline of the brainstem, you will lose this ability to synchronize the jaw movement. You need some sort of connections across the midline at the brainstem for bilateral synchronized jaw movement.
Fan Wang. From the nervous system to the skin now, and the issue of eczema and whether it's linked to skin cancer. KCL scientist Esther Holster, and before her, Fiona Watt.
There's been a long debate about whether or not people who suffer from allergies have an altered risk of developing cancer. Looking at the figures, I would say for every study that suggests a positive correlation, there's one that shows a negative correlation and one that shows no effect at all. But nevertheless, it is of interest because there's a very strong emerging concept that harnessing the immune system in different ways can be a useful way of treating cancer.
Why should there be a relationship between dermatitis and skin cancer?
In theory, if you believe that the immune system is involved positively or negatively in tumour formation, then we know in eczema that the immune system is on red alert, and that's why you have dry, scaly and itchy and red skin. It's quite natural to expect that in that situation you would have an altered response to insults that would cause cancer.
And so one hypothesis would be, if you have an immune system on red alert, that it should be more vigilant towards any cells that are trying to become cancerous and delete them, but then is not the flip side of the coin that an immune system on red alert is going to cause more inflammation, and we know that sustained inflammation is in and of itself a risk factor for cancer.
Yes, absolutely. When we set up the experiment, we were completely divided about what the outcome would be. We genuinely didn't know what to expect.
Esther, you work with Fiona on this project. How did you actually do the study to try to unpick which of those two outcomes it was going to be?
Because the human system is very difficult to study, because atopic dermatitis or eczema is a disease which has a relapsing and remitting nature, which we had to shift to an animal model to study the importance of the atopic phenotype on skin carcinogenesis. Because people that are suffering from atopic dermatitis are often treated with immunomodulatory drugs, and that actually will clearly hamper our conclusion. So we created a mouse model in which there are 3 proteins lacking that play an important role in the epidermal barrier function.
And does the absence of those proteins then trigger an atopic state in your animal model?
Yes, so if you knock out those 3 proteins, what you get is you get an increase in certain immune types, while other immune types are actually reduced. And phenotypes that we see are actually, they exhibit hallmarks of the active state of atopic dermatitis.
So this gives you an animal where if you were to look at the skin, you would see characteristics very reminiscent of atopic dermatitis in a person.
Yes, that's right.
And so you can be reasonably sure that you've got a faithful model there in which to then test the rates of cancer development.
Yes, I think that's one of the biggest novelties, that we used for the first time an animal model in which the epidermal barrier is clearly defective and has also an immune component which really mimics the disease.
And then what about the skin cancer side of things? Because animals don't develop skin cancers over the lifetime like a human does necessarily. So how did you mimic that?
Yes, so in order to induce tumor formation in these mice, we make use of two chemical components. One is a component called DMBA that actively induces mutations in HRAS, which is a very important protein in tumor formation. And if you just apply this DMBA agents, mice don't get tumors. But if you promote the tumorigenesis with another component that is called TPA, you start to get tumors.
And so if you then do the obvious experiment, which is you do this in mice that have the deficient skin barrier, so they're mimicking atopic dermatitis, eczema, versus animals that don't have the deficient barrier, so they're like a person who doesn't have eczema, what's the difference in rates of skin cancers in those two groups?
What we saw is that the mice that have atopic dermatitis, they're highly resistant to skin tumor formation, so they develop much less tumors. And there were also mice that did not develop any tumors at all, while the control mice all made tumors.
Quite striking findings there, Fiona. How do you actually account for this, and what do you think the implications more broadly are then?
I think in terms of the basic biology, the important point is that if the skin has a defective barrier, in our model, that is linked to increased cancer resistance. And we believe that that is because the immune system helps to ensure that cells with cancerous mutations are expelled from the skin. So that's the key conclusion from our findings.
Fiona Watt and Esther Holster. You're listening to the eLife podcast with me, Chris Smith. Still to come, the proteins that move molecules around cells, and eLife's editor-in-chief, Randy Schekman, on why the present mainstream system of science publishing is, in his view, deeply flawed. Before that, to trypanosomes, the organisms that cause sleeping sickness. They're extremely difficult to eliminate from the body, partly because the organisms frequently shuffle the antigens that they express on their surfaces, so they present a moving target to the immune system. How this antigen shuffling trick is achieved, though, is a mystery. But now the University of Würzburg's Christopher Batteram has found that when the bugs activate a new antigen, they use an epigenetic system to shut off the expression of the old one.
African trypanosomes, which cause the sleeping sickness in humans, evade the host immune system by expressing markers on the cell surface that are recognized by the immune system. They have a huge family of these marker genes in the genome, but only one is expressed at any given time. Nothing is known about the mechanism of how this is achieved, how just one marker is expressed at any given time and the other ones remain silent.
Do the parasites, the trypanosomes, change those markers at various points in order to make themselves look different, give themselves a chemical facelift, if you like, so the immune system finds it hard to recognise them?
Yes, they periodically switch the expression from one marker to another. It's not known how they do these switches.
So the problem you've got is, how do we turn off a very big group of genes, leave just one active, but then periodically totally mix things up and select a gene from the inactive group and suppress the one that was turned on?
Exactly. The question was how the trypanosomes activate a new antigen and how they deactivate the previously active one. One possibility is that the active one could first be silenced before a new one is activated. There's a little problem with that because silencing the active gene by RNAi causes a cell cycle arrest and the cells eventually die. So we hypothesized that this could be the other way around. A previously silent gene is randomly activated and thus this causing the deactivation of the active one.
I see. So a new one switches on, and it's the process of switching on the new one that in some way turns off the existing one. And in this way, you've always got a surface marker gene active, so you won't go into cell cycle arrest, but you don't turn off the existing one until you've got a new one ready to go.
Exactly.
What experiments did you do to then work out whether that was the case? Did you put some kind of marker into one of the genes and then turn it on with another to see if that marker does what you would predict it would?
Yes. So we introduced a second surface marker gene into a different genomic context, which we can inducibly express by adding a chemical.
And when you turn on that second gene, does the first gene switch off as you would predict it would?
Yes, it does.
How did you then unpick that? So do you know what the new gene coming on is doing to the transcriptional system for the first marker gene to make it shut down? How's that being achieved?
So this remains unknown. We know that it depends on histone modifications.
So that's interesting. So it rather than it directly shutting off promoters or something, it's doing it via a modification of the histone. It's doing it epigenetically.
Yes, exactly. That is the case. We did the same experiments with a knockout cell line in which a histone methyltransferase was deleted. And when we overexpressed the second gene in these cells, then the endogenous gene was silenced, but not the whole transcription unit.
So you overexpressed just the surface antigen gene for the second surface marker, and this had this shutting down effect. So there must be something intrinsic to that antigenic signal which
Yes.
is activating this epigenetic modification apparatus.
Yes.
How is
exactly.
it doing that?
We don't know. This is the big question we are challenged with now.
Would you like to speculate, because you must have some theories, as to how it's doing this? What do you think's going on?
It could be that the initial spike of the production of two surface antigens could relay a signal to the silencing machinery in order to maintain this monoallelic expression. So one possibility is that there's a signal coming from the endoplasmic reticulum, a folding protein, so-called chaperone or something like that, but we have actually no idea.
And of course,
Otherwise,
this could be very important, this mechanism, because one of the big problems with sleeping sickness, trypanosomiasis, is that it's very hard to rid the body of these bugs. The immune system can't get a handle on them because they evade immunity. So if we can find a way to clog up this system and stop them shape-shifting themselves in this way, maybe the immune system would gain the upper hand and eliminate them.
Exactly. This is the future goal, to get more insights into this mechanism, how the transcription unit is silenced. And when we can trigger this chemically, then we can get rid of the parasites, maybe.
Christopher Bartram. Now to the cellular postal system. Margaret Robinson and Jenny Hurst, working in Cambridge, have between them invented a new electronic tool to probe various databases looking for previously undiscovered proteins that form complexes that move molecules around inside cells. And what their system flushed out was a whole new complex, TSET, T-S-E-T, that can trace its origins back to the first eukaryotes. Margaret Robinson.
So what we're interested in is how different molecules find the right part of the cell. And we're interested in the machinery that the cell uses to get things to the right place. And we're especially interested in things called AP complexes, which stands for adapter protein complexes. Each of these complexes is made out of 4 different proteins. And for many years, we thought that there were 4 of these complexes. And then just a couple of years ago, we discovered a 5th, which took us all by surprise. And we thought, well, maybe there are even more of them out there.
And their role is quite literally to package things up to the right place and address it to the right part of a cell.
Exactly. What they do is they find molecules, they package them into little containers that can then be delivered to a different part of the cell.
And Jenny, if you want to find something and you don't even know it exists, which is what you had to do with this study, how do you do that?
First, you scratch your head and you try and think of some ideas, and very long conversations over dinner with my husband. I suspected that there was something else to find, but it was a case of putting our heads together and using some computing skills, because the 5 complexes we already knew about, we knew what the family resemblance was, and it was a case of figuring out how you were going to use that information to search hundreds of thousands of protein sequences, but automating it.
It's a bit like Mendeleev with his periodic table, and he knew there were gaps that would fit the board, but didn't have the element for them at the time, and then knew where to go hunting. So what did this show, Margaret?
So what we found was we know that there are 4 subunits in each of these complexes, and what Jenny discovered was 1 new member of each of the 4 subunit families. So we had the 4 subunits that might be used by a cell to make a whole new complex. And the next step was to find out whether these 4 proteins really were all stuck together. And for this, we couldn't look at humans because although humans have got these 5 AP complexes, the 4 proteins that Jenny discovered with her new tool didn't all exist in humans. They existed in plants and slime molds and this weird soil amoeba, but animals have lost 3 of the 4. We've hung on to the 4th one, but not the whole thing, just a little fragment of it.
Do you think there's any possibility that because Jenny's tool is mining data that's already been published by people, that the undiscovered has remained undiscovered and that people just haven't published the protein sequences that, were they to exist, her tool would have found them?
I don't think so, because it's not just publications, it's things like the entire human genome. So we know the sequence of the whole human genome, and from that we can predict the sequence of every protein in humans. And the same is true with lots of other organisms, including plants and these weird things like the slime mold and the soil amoeba. So I think that the proteins are there in the database. It's just a case of finding them.
And what are the implications of, now you've got these additional proteins, admittedly not in us, what can you do with this information?
It's more about understanding where these important proteins that we have today, where they came from. So I suppose it's a leap to think, well, we're looking at something that's today and in disease, but I think it's important to know how these families evolved.
Yes, I think it's about our understanding of the evolution of life on Earth. Especially since this new complex predates the 5 that we already knew about. It must have existed long before the last eukaryotic common ancestor, 1.5 billion years ago. We're talking more like 2 billion years ago, and it tells a lot about how cells like our own actually evolved from much simpler cells way back then.
Margaret Robinson, and before her, Jenny Hurst. Finally this month, a word from eLife's editor-in-chief and Nobel laureate, Randy Schekman, who explains why, in his view, the present commercial system of science publishing is distorting science. And he also sets out his vision for what eLife is doing to change things.
I felt very strongly that, particularly at the high end, in the life sciences, young investigators felt undue pressure to publish in a very small number of venues, commercial journals such as Nature and Cell and Science Magazine. These are fine journals, they publish important work, but they artificially restrict the number of papers and pages that they publish because, at least in the case of Science and Nature, they're still modeled on a print version, and it's expensive for them to print more pages. And so they've created, I feel, an artificial commodity. They do this, frankly, to sell magazines. And this has, I believe, a distorting influence on the way young scholars choose to publish their most important work. They compete for limited resources, even when those resources represent an artificial commodity. In the 21st century, it seems silly to me to be making decisions based on a print run when most young scholars read everything online. They don't even know these journals exist in a hard copy form.
But you were editor-in-chief of PNAS, the Proceedings of the National Academy of Sciences, another very prestigious journal.
Yes, I was, and that's actually where my opinions formed most clearly. I saw that in spite of the success of the proceedings, that investigators still preferred to publish in venues that restricted the number of papers that they published. At PNAS, we felt that it was important to publish all the great papers that we received, so we didn't have such an artificial restriction. And yet, scholars chose not to send their best work generally to the PNAS because of the influence of a number called the impact factor. It's a number that greatly, I think, influences decisions about where young scholars prefer to send their work because they feel review committees, grant committees often base their decisions on the impact factor of the journals in which work is published. And these journals, particularly the commercial journals, use that number shamelessly in their advertising. Indeed, they manipulate the calculation to exaggerate the number and to give themselves even more influence in decisions about where scholars publish.
And you're seeking to change this with eLife?
Absolutely. We will not restrict ourselves based on some perception of the number of citations that a paper will generate or the number of papers that we publish. If we get great papers, we'll publish all the great papers we evaluate.
The other key difference, of course, is that if you publish in those aforementioned journals, in order for me, if I send them a piece of work, as one person told me, they were grossly offended to be told by the journal to go and buy a copy of their own paper when they wanted a reprint of it, because that's how these journals make money. They're selling the science that the taxpayer funds to the scientific community.
Absolutely. They're in the business of selling magazines. But the fact is that they make money on both ends. They make money selling magazines, they make money on subscription licenses, they make money from advertising, and they rely on the academic community to do essentially most of their work in evaluating the science, who are not compensated for their services. I think scientists have bought into this inappropriately.
So what would you like to see change, and how will you make a difference?
Our challenge is to capture the imagination of scholars, having a venue that they will seek out to publish their best work. So we have to influence young people who are at a crucial career stage so that they make those decisions based on how their work is seen by the community and not based on some artificial number like an impact factor. The other thing I want to say is this impact factor is influenced not necessarily only by the quality of the work, but by whether the work is fashionable. And I'll give you an example. Nature published 2 papers a couple of months ago where the authors claimed that they could convert adult cells into embryonic stem cells by a simple low pH treatment. The work was met with great skepticism. In fact, people are unable to reproduce these results. There was evident manipulation in the papers, and now people are quite suspicious. But the fact is that these papers will generate thousands of citations for Nature, and so they will profit by having published those papers, even if they end up being retracted.
You're getting behind an initiative called the OA, or Open Access Button. What is that, and why are you putting your name to it?
Yeah, so it's a very interesting program, just beginning, to identify when a reader has trouble getting behind the firewall of a commercial journal. For those people who are aware of the Open Access Button program, you can automatically alert this service when you have failed to gain access to published work because of firewall restriction.
But how will you use the data that comes from it in order to enact change?
Well, I think it'll be very striking to realize with the data that they compile how many people are frustrated by their inability to gain access to the literature. Commercial journals may be forced inevitably to introduce open access journals on their own. In fact, I know that the editor of Nature, Phil Campbell, has indicated that he sees the open access movement as the future, and he suggested that maybe that even Nature may eventually go open access, but they feel that they'll be able to charge. He suggested, for instance, $30,000 might not be an unreasonable sum to expect.
And of course that money would have to come from the scientist, which inevitably means it would come from their grant pot, wouldn't it? The money that the taxpayer in some cases, or a charity like the Wellcome Trust, gives to the scientists to do their research.
That, of course, is the problem, and the question is, would people be willing to pay that sum of money to publish their papers? I should tell you there's another undercurrent that has taken shape in Asia that I think is quite alarming, where scholars in China are literally offered bribes to publish in these high-profile journals. Last year, the Chinese Academy of Sciences issued a bulletin to its scholars offering a cash reward of the equivalent of $33,000 for success in publishing a paper in Cell, Nature, or Science, irrespective of the subject matter of the paper. So you could well imagine that an author, having won the lottery and published in these journals, would simply take the profit that the Chinese Academy of Sciences offers to pay the fee that the journal charges for an open access version of the paper. I think this is a terrible distortion in the system.
UC Berkeley's Randy Sheckman, eLife's editor-in-chief. That's it for this month. Don't forget that all of the papers we've been discussing are freely available alongside previous editions of this podcast from eLife's website. That's at elifesciences.org/elife. My name's Chris Smith. This is a Naked Scientist production. You can find out more about us from our website at nakedscientist.com, and I'll be back next month with another look inside eLife. Until then, though, thank you for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. online at elife.elifesciences.org.